This presentation is about the grape flower, and I have some other flowers as, as examples, excuse me. So I have a PowerPoint to start off on the grapevine flower. And we'll get going here. Grape flowers look a little bit different than your classic flower, and they're a little bit different terminologies, which I'll talk about, but very little. Most species of grape in the wild have a separate male and female flowers on separate plants. And so if you have male and female plants separated, you call that a dioecious plant. Di meaning two, and dioecious plants are separated with male plants and female plants. But the commercially important plants in the Vitis grape genus are monoecious. Commercial varieties are typically monoecious, meaning the male and female flowers are on the same plant. That's specifically monoecious, but they have perfect flowers, meaning that the male and female parts are actually in the same flower. So when you're talking about an ecologist or a biologist, taxonomist, somebody who's interested in the broad range of grapes, most of them are dioecious, but as a vineyard manager, somebody who works in the commercial grape industry, it is monoecious or perfect flowers. Now, not all monoecious plants are perfect. Something like squash. Squash is monoecious, but they have male and female separate flowers. And they're pretty easy to spot if you ever have some squash, some zucchini or summer squash, and you look at the different flowers, you'll notice that there are definitely male flowers that are a bit smaller, and they have the large anthers with lots of pollen, and the female flowers don't have that at all. They have a stigma and a larger swelling at the base that's going to develop into the fruit. Most of the pollination in grapes is wind pollinated. The vast majority, there's some evidence that insects are involved, but it's probably a statistically insignificant process. The vast majority is wind, with some very minor pollen transfer that occurs here and there on occasion due to the activities of insects. And because of the way the wind pollinates, it shakes a flower, so the vast majority of pollen is transferred from the anther, the male part containing the pollen, directly onto the stigma in the same flower. If it's not the same exact flower, the pollen isn't often carried very far. It's probably on the same vine, even if it's a different flower somewhere else in the cluster or another cluster right nearby, it's still self-pollinated if it lands on a flower on the same plant or that is clonally identical. So in a block in a vineyard, you would call all of that technically self-pollination because they're all clonally identical plants in a block. But usually when we think of self-pollination, it's mostly the same flower, pollen from the anther on the same flower goes to the stigma on the same flower, or a nearby flower on the same plant. After pollination, step one, the pollen from anther to stigma, it takes about 2 to 3 days for fertilization, because inside of every pollen grain is 2 sperm cells. And so there's a double fertilization. You could look it up. I'm not going to go into it here, but one sperm cell fuses with the endosperm tissue, one sperm cell, and fuses with the egg inside the ovule. And there's different reactions that together make the seed. It's an odd situation in plants where they have what's called double fertilization. Look it up if you're interested. I'm not going to ask you about that on a test. But the sperm has to be delivered, and one sperm cell must unite with one egg cell in the ovule down inside of the ovary. You can see here the ovules. There are multiple ovules in each ovary. Each one of the ovules will develop into a seed. And as you should know, if you've eaten very many grapes, that if they're not seedless grapes, there's more than one seed in there. Often 4 seeds is not too uncommon. And that process, the very initial process of the sperm cell fusing with the egg cell, is called fertilization. And the pollen has to grow a pollen tube. The pollen is a living organism, and it grows down through the style and until it reaches the ovule, and that takes a couple of days to grow down and deliver that sperm cell. Water causes the calyptra, the fused petals, to be retained. So it can wash away any stigmatic fluid as well. So you don't want the calyptra to stick around. You want the calyptra or cap to fall, releasing all of this to the wind and the pollination to occur. And you want the pollen that gets on the stigma to have time to germinate the pollen tube and start growing and deliver those sperm cells. So water, either through sprinklers or rain, is a bad thing during flowering because it causes problems with the fused petals called the calyptra, and it causes problems with washing away the pollen. And so you end up getting lower fertilization, lower pollination, and a reduction in your yields. And it can be very severe if you have a bad rain event right at the wrong time. You can have some pretty sparse looking clusters. The calyptra, or cap as it's called in the industry, the calyptra is fused petals, and they're fused at the tip. They typically are greenish in color, or a very, very, very pale green where you might at a distance think it's white. And they are fused. There are 5 stamen with a stalk. At the base of each filament are special nectaries that produces a scent that can attract insects. Is that really important? Well, since they're wind pollinated, maybe that's something that is evolutionary holdover, kind of an unnecessary thing that the flowers are doing since they don't need the insects. Or maybe there is some benefit to having those active nectaries. I'm not really sure. Sometimes we think something's not important, and you look at it long enough, study it hard enough, and you find out, hey, it does serve a purpose. The stigma does have some fluid. It is wet, sticky with nutrients and sugars, food to feed the growing pollen. The ovary at the base, this portion here is the ovary containing the ovules, and 4 ovules each containing an embryo sac. Don't worry about the pericarp. There's some details here, don't worry about it. If you understand the calyptra or cap, same thing. You understand the fact that we have typically monoecious or perfect flowers, you're doing all right. And you understand what an anther, stigma, style, ovule, egg, you know, the basic things that I've already mentioned. There's a lot of variety in grape flowers, and here you can see at the top some of the wild ones, the male flowers that don't have a functional ovary inside. Now they're just showing one of the anthers with the, or actually one of the stamen, the anthers and filaments, but there would be 5 of these typically. And then at the bottom are the female flowers. Even though they do appear to have stamen and anthers, they're dysfunctional. They're there in a developmental capacity, but they're totally underdeveloped and they don't produce viable pollen. And so they're dysfunctional female flowers. The perfect flowers have both the female and the male parts. So again, there's a lot of variety out there, but not when we're talking about commercial grapes. I don't care if you're talking about wine grapes, table grapes, raisin grapes. Commercial grapes are almost always perfect flowers. Inflorescence development, the growth of the inflorescence up to and until just after anthesis is dependent on stored reserves and must compete with shoot growth. So there is a process called the source and sink. The source is where the sugars or food is coming from. Primarily during this season, this part of the season, it's mostly coming from active photosynthesis in the leaves. That's the source. And then the sink is any place that is collecting those sugars, where the sugars are flowing to. Just like we have a sink. Why do we call it a sink at home? To wash our hands, wash the dishes. It's where the water flows, where the water collects and goes to. Well, in sugars in plants, the sink is where the flow of sugars is going. And based on different metabolic factors and physiology of the plants, there are two major sinks during flowering. The flowers themselves going, hey, we need sugar, we need to grow, and the growing tips of the shoots. And the growing tips of the shoots are actually really strong sinks and generally outcompete the flowers. And doesn't mean the flowers aren't going to get any, but the majority of sugars, a slight majority of sugars, is going to canopy development as opposed to flower development. And there are a lot of reserves that are necessary that are being shipped to the inflorescence. And these stored reserves of sugar, as opposed to the source sugar, the new sugar from the leaves, is critical in the early stages. But once those reserves are used up, once the reserves that are kind of in waiting for the inflorescence development to occur, once they've used up that, now they're in direct competition with the shoots and they need new sugars. Poor fruit set can also be caused by poor growth in the previous year, because what happens in the previous year, that's where your stored sugar is going to come from. You've got stored starches in the roots, and plants store sugar as starch. And so the plants put all of this starch in various locations of the vine. And if you had a bad year last year, then no matter how good photosynthesis is this year, you're still short on those starch storages that can be disassembled and turned back into sugar. You don't have the starch, you don't have the storage, you don't have sugar reserves. So poor growth last year. Also overcropping, if you overdo it. And I guess that's one of the reasons why people have these pounds per acre limits. It's not just about quality this year, it's also quality next year. If you really push your vines and go, wow, I got an extra 3,000 pounds per acre off of my block this year, you just may have done some damage to your potential growth and fruit set next year. Leaf loss due to whatever reason, stresses, pruning improperly, animals, insect damage, whatever, moisture stress. So some varieties are much more sensitive than others, such as Merlot being very dependent on the previous year's stores, Pinot Noir, not so much. So you would expect there would be a variation, because there's a variation in every characteristic we ever look at. Different varieties behave slightly differently. And so these storage reserves are very important in Merlot, much less important in your performance of Pinot Noir. I already mentioned this, the shoot apex or the tip of your shoots growing, the apical meristem is a stronger growth sink than the inflorescence. So it's going to really have a strong attraction for those new sugars. And therefore, vigorous growth is going to take away even more sugars from your inflorescence development. How do you get in a stronger, more vigorous growth? Either through your rootstock selection, which is crucial. You can't change that, or your fertility practices. You can change that. If I give a little too much nitrogen in particular, I'm stimulating vigorous canopy growth at the expense of inflorescence growth. You can do tipping of these shoots at flowering, and by doing that, you essentially have taken away those apical meristems at the tip of the shoots. And that's temporary, but if it's timed right, you tip it, and now all of a sudden that auxin development and the growth of those apical meristems making more canopy is slowed way, way down. After a delay, after a couple of weeks, they'll start to kick in with lateral growth from the axillary buds, and they'll start to become strong sinks. The axillary buds become their own apical meristem tips, making new branches. But there's a delay from the time you do your pruning. There's a couple week delay before you start getting another strong sink pull in the axillary bud development. So tipping is a great way to help divert sugars into your developing inflorescence to get good fruit set. Again, some varieties in certain climatic conditions might benefit greatly from that. In other climatic conditions or other varieties, maybe you're wasting your money doing tipping. The basal leaves are essential in the development of the inflorescence. A lot of those basal leaves, they're the nearest leaves to your clusters on most varieties. It is the clusters develop in the lower portion of the cane or shoot, and the basal leaves are nearer and therefore more important in the pull of the sink of those inflorescence, developing inflorescences. And so the basal leaves must be healthy, they must be present. And so you definitely want to be doing canopy management right. If you pull too many of the basal leaves, you're leaving the upper leaves, which are even closer to the tips and a stronger pull away from those inflorescence. Certainly nitrogen deficiency will cause a problem in flower development, but that is very variety dependent. I also mentioned just a moment ago, it's not on the slide here, but nitrogen excess causes a problem too, because a nitrogen excess creates an artificially high vigor in the development of the vines. The vines start pumping out more leaves instead of fruits. Doesn't seem to be a very strong correlation with the others, the phosphate, the potassium, calcium, magnesium, sulfur, in terms of flower or fruit set, I should say, or number of flowers. Now, certainly you need enough of them. You need enough of all 6 of those macronutrients, but there isn't a strong relationship there. Boron is needed for germination and growth of the pollen tube. Boron is hugely important in flower and fruit development. And as I think I've mentioned already in this class, the difference between having too little boron, deficiency, and too much boron, toxicity, is a small margin. You've got to be good, accurate with your available boron that is appropriate for your crop. Zinc response varies by variety and location. Sandy soils are the worst, as you might expect. Sandy soils are the worst for everything when it comes to holding on to nutrients. Zinc does increase seed number, which can increase berry size. which again relates directly to yield. So it's important to have a consistent history of how you fertilize, because a lot of what we're doing is trying to estimate yield weight, and we know yield weight based on cluster uh, number, but then we're guessing at what our cluster size and cluster weights are going to be. And if we keep changing our fertilizer routine, trying to tweak things, we might have the same number of clusters per acre this year from last year. But if we're altering zinc, we might change the number of seeds, which increases the cluster weight, which might have an impact on our overall yield weights. Again, a lot of these estimates we do are dependent on being consistent with our practices. But a lot of learning is doing things differently, trying new things. So there's a balance there between being consistent and being innovative and learning and researching. Molybdenum. Molybdenum has a strong role in nitrate reductase. So in order to use nitrogen, you need molybdenum. Therefore, without adequate molybdenum, you can't utilize nitrogen, and there are all kinds of problems with that. Extreme moisture stress at bloom is bad. You definitely do not want to dry out your field at that time. You want your vineyard to be happy. Pollination. Pollination is released at cap fall. Again, one of the reasons why rain is bad, it holds onto the cap after a rain. The cap doesn't fall off, and you're not going to get pollination occurring properly. And then if it happens after cap fall and you get a rain, then you rinse off all your pollen grains. Pollination prefers warm, dry weather. If it's really humid, if it's really cold, if it's really sticky, it doesn't like it. Rain is bad. Wind is bad. You need some wind. You need it to be a breezy and an occasional afternoon period of a few gusts is not a bad thing at all. But you don't want heavy winds, and you don't want sustained winds, because that's going to shake too much of the pollen off, and it's going to get caught in the wind and carried away from the vines rather than landing on the stigma where it is going to be able to grow and fertilize a couple days later. You can actually make good solid estimates of your yield estimates, your indirect measure of how well you're going to get fertilization and fruit set based on pollen counts. And the ag industry, as well as the medical community, as well as air pollution control districts, all are very carefully monitoring pollen for various reasons. And so you can look at what is the average pollen. Doesn't matter what pollen, just the average pollen counts in the air can tell you what kind of fruit set you might expect in your fields. And if you collect this data for a while, read up on some of the literature in this area, and you can start making a correlation. What pollen counts are correlated to a good fertilization, good fruit set, and what kinds of pollen counts might you expect for a poor fruit set? Because the pollen counts are going to go up and down for the things exactly like we're talking here. Pollen counts go way down in the rain. Pollen gets carried out of the air. Low pollen count, low fertilization. With high or low temperatures, excessively high or excessively low temperatures, pollen counts will go down at the extremes because plants aren't happy and pollinating as effectively, or shedding their pollen as effectively. High humidity causes pollen to become sticky and less likely to be in the general atmosphere. So pollen counts go down in extreme humidity conditions, which also hurts fertilization. Dry, warm, but not too warm. What else do I want to say here? Most varieties are self-pollinated. I've already mentioned that. Pollen germination optimal at 26 to 32 degrees centigrade, which is warm, but not too hot. What is normal human body temperature? 37 centigrade or 98.6. That's way too hot when you get up there into the 90s. So this, I don't know off the top of my head, but we're talking about in the upper 70s, 80s, they're happy for daytime temperatures. You start getting closer into the 90s, you got a problem, and you start getting really chilly. 15 is getting pretty chilly. It's not going to be good for pollination. I already mentioned boron and calcium fertilization. Pollen grain lands on the stigma. It absorbs nutrients on the stigma. It has food and nutrients and starts to grow if it has the right molecular signature. There's all kinds of almost chemical warfare going on. If it's the wrong pollen grain from an incompatible plant, that pollen grain either won't get the signal to germinate or it will be killed if it tries to grow. And that's to protect it from having the wrong pollen, wasting the nutrients on that pollen tube growth, the nutrients in the style and on the stigma, and wasting all of that. So the stigma has this chemical warfare to prevent the wrong pollen from germinating. And then there's additional chemical warfare going on as the pollen tube is growing. If it's the right pollen, the style, all of the cells of the style act to support and feed that growing pollen tube. If it's the wrong chemical signature or molecular signature of a pollen tube that did successfully grow into the style, the cells of the style will attack it and try and stop it from growing any further. Once it reaches the ovule, the pollen tube grows into the ovule and delivers the sperm cell to the egg inside of the ovule. The two join, the egg cell and the sperm cell become a single cell called a zygote, and that process of two cells becoming one is called fertilization or syngamy. I don't use that word in this class, but in plant science we use that word, syngamy, S-Y-N-G-A-M-Y. S-Y-M, or N, excuse me, S-Y-N, N as in Nancy, G-A-M as in Mary, Y, syngamy. Don't worry about it on the test, but hey, this is more of a upper division class. You should start learning these important biological words. Temperature is very important. Again, warm, dry, the same conditions that are good for the pollination is good for fertilization. If a flower isn't fertilized effectively, then the flowers will drop. And we call that shattering. You get this massive amount of these tiny flowers. It almost looks like somebody sprinkled confetti under all of your vines when shattering is occurring, because all of these tiny little flowers are dropping. And you need shattering. There are way too many flowers on each cluster. If you had a cluster with 100% germination, or excuse me, fertilization of all of your flowers, and every flower developed a grape, you would have way too many grapes in a cluster. It wouldn't work. But the fertilized ones remain on the cluster and develop. The unfertilized ones can occasionally remain on the clusters as well. And we call those millerundage. Millerundage. And that is the tiny little bitter unfertilized grapes. When you look at a cluster of grapes, a bunch of grapes, you might call it hen and chicks. It's when the berries of different sizes and levels of maturity are on the same cluster. The normal-sized seeded berries are the hens, and the small seedless berries are the chicks. And it's not a good thing when you have a lot of chicks. You want hens, and the occasional chick in there is not a big deal, but if you've got a lot of them, there was a problem of some sort. To get the problem where you would call it hen and chicks, you don't use that term unless it's a problem. If you're saying, hey, I've got hen and chicks in my clusters, that's probably caused by cold weather during flowering, and possibly other factors that are impacting proper fertilization and proper shattering during that crucial fertilization period. And you get this bizarre hen and chicks. You might also hear other terms depending on where you are. It's kind of regional, since this is in the field terminology, not biology. Shot berries, pumpkins and peas, and the French term, which is the most, I would say, widely acceptable term, the millerandage. Now, millerandage isn't talking about, or shot berries are not talking about hen and chicks. The shot berries and the millerandage are just the chicks. Pumpkins and peas are the hens and chicks. Fruit set. Fruit set is a transformation of flowers to berries. And so once you have a fertilized fruit and it sets, it is going to develop into a berry if it's developed normally. As I mentioned, clusters can have a lot of flowers, up to 1,000 flowers. That's a very, very full cluster of flowers. And 70 to 80% of them typically drop off. And that's normal. During shattering, you lose 70 to 80%, and they'll just shrivel up and fall off. And fruit set is a very simple calculation. It is a percent calculation, and it's equal to the number of berries that you have divided by the number of flowers that you have. And if you just do berries divided by flowers, you'll get 0 point something. You'll get a proportion or a decimal number, and you multiply that times 100% to turn it into a percentage. So how many berries do I have and how many flowers do I have? Well, you just got to count and estimate. So during flowering, it's important to measure this. It's important to take an estimate of what is my flowers per cluster. That's one of the data points you collect during the growth period. And then certainly later, when you get fruit set, you do another count. What is my average number of berries per cluster? And then you can know what your fruit set percentage is. 20 to 30% is certainly normal, but the bigger the inflorescence size, the more likely you're going to have a lower number. You can't have huge cluster size. If you start off with huge numbers of flowers, more of them are going to shatter. If you start off with smaller numbers of flowers, less of them are going to flower. But variety, cluster size, and other factors are all going to be important in terms of what your fruit set percentage is, as well as what happened this year during flowering and fertilization. Once you get pollination and fertilization, boom, that's the trigger. That's the start of berry growth. But it's not going to continue unless the fruit properly sets and stays as part of the cluster. Cell growth, not going to worry about that. There's an abscission layer that is at the base. There's a layer at the base of the pedicel, the base of the flower stem, that joins the flower to the rachis. That's a little pedicel. And that little flower stem that joins it to the rachis, or the cluster stem, the rachis, there is a point called the abscission layer. And if there is not proper fertilization, 2 layers of cells grow, one at the base of the pedicel and one on the rachis, on the stem. And if those 2 layers of cells develop, they're like a perfect sealing off point and the flower falls. If you do get fertilization in that flower, then that fertilization sends a signal to stop the development of the abscission layer. Those two layers of cells between the pedicel and the stem don't develop, and the flower stays affixed, and as it grows, it develops into a strong attachment point. So that's what we're talking about with the abscission layer. It's exactly the same process with leaves in the fall, except all of leaves on deciduous trees that are healthy get the signal for the abscission layer to form. And so right now, abscission layers are forming like mad out in the field because it's fall. And so at the base of deciduous tree leaves, at the base of grape leaves, included as other deciduous plants, they're forming deciduous abscission layers. When the temperature and other conditions are right, the abscission layer will form and complete. The leaves will turn red or yellow or orange as they're being stripped of nutrients. Once the nutrients are stripped, the abscission layer finishes and the leaf falls off. Same process. So this process of fruit set after fertilization is about a 2 or 3 week process. The percent fruit set depends on the bunch number per vine. So that's certainly your yield component compensation, the yield that you have already established based on your pruning, the bunch size, each cluster size, the variety, the climate, and the rootstock. All of those things together will influence fruit set. So that is it for the grape flower. But in addition, and I'm not going to have separate PowerPoints for this, I have a drawing that you can use with some important words: calyptra, cap, and thesis. These are important vocabulary words, and a great picture of a grape flower, a commercial grape flower, and the calyptra or cap falling off, opening it up, preparing it for pollination and fertilization. I also have a malice or apple flower, just as comparison. And you can see this inferior ovary. Inferior means it's just below the point of attachment of the petals and other whorls of the flower. So all of these parts of the flower are joining right here, and the ovary is below that. So we call it an inferior ovary. But just for comparison, that's an apple. Here's a cucurbit, a male cucurbit flower in a microscopic prepared slide. So this is the undeveloped female portion, but here are the stamen with the anthers, and you can see the pollen inside the anthers. All of these kind of deep burgundy dots represent the pollen. So that is a male cucurbit. Here is a similar type of a slide preparation showing a self-pollinated tomato. There is a staminate cone. So the stamen produce what would be a cone. The stamen are surrounding the stigma and style. And as the stigma is being pushed up, as it's growing through the staminal cone, it picks up pollen on it. So it's fertilized, or not fertilized, but at least pollinated before the flower even fully opens, or certainly within a day after it opening. Again, it depends on the variety, but it pollinates itself very quickly as a self-pollinated crop. The cucurbits, they're separate male and female flowers. They need pollinators like bees to be crawling around, feeding inside the flowers and spreading the pollen as they feed. And that's it for flowers.